Hello, and welcome to the AMP Nanostring Corporate Workshop. Thank you for joining us today. I'd like to go over the agenda for today's session, and we'll start with a digital spatial profiler overview given by Nick Conforto, Senior Director of Global Marketing at Nanostring Technologies, followed by a quick set of polling questions, and then we will hand over the scientific presentation to Dr. Christopher Corless, Professor of Pathology, School of Medicine, Vice Chair of Molecular Pathology, and the Director of the Night Diagnostic Labs at Oregon Health Sciences University. It's my pleasure to introduce our two speakers, Nick Conforto, Senior Director of Global Marketing at Nanostring Technologies, where he is responsible for leading the development and execution of the strategic plan for the digital spatial profiling business. Followed by Dr. Christopher Corless, Director of the Night Diagnostics Lab at Oregon Health Sciences University. Dr. Corless received his MD and PhD from Washington University in St. Louis, Missouri. He did his residency training in anatomical pathology at Brigham and Women's Hospital in Boston, Massachusetts, where he also completed fellowships in gastrointestinal and genitourinary pathology. Dr. Corless is a board-certified anatomical pathologist, and he joined the faculty of Oregon Health and Science University in 1994 and became a professor of pathology in 2004. I'd like to hand the presentation over to Nick Conforto. Thank you.
Thank you, Cindy. Yes, pleasure to be here virtually with you all today. Looking forward to providing a quick brief update on our market-leading spatial platform, our GeoMx digital spatial profiling technology, and how it's now being utilized from a translational setting, to now be able to do clinical trial work and clinical testing and validation on clinical samples for spatial profiling. And then I will hand it over to Dr. Corless, who will share some of the most recent data that they've been able to generate in his lab there at OHSU. So for those who aren't aware of who Nanostring is, we are based in Seattle, Washington. We were founded in 2003, and we develop tools that enable translational research and precision medicine work. So that started with the launch of our nCounter system. Many of you may be familiar with it. That is a gene expression platform that allows for multiplexed gene expression using direct hybridization chemistry to do that in a very simplified, automated manner. We launched our FDA-approved ProSigna test with that technology, and to date we have a few hundred boxes worldwide, and that install base continues to grow and continues to be utilized. And in 2019, we launched our first spatial technology. So while the nCounter was mostly used for bulk gene expression work, the GeoMx system was our first step into spatial biology and understanding spatial heterogeneity. So that launched in 2019, and that has been the market-leading platform ever since to do spatial expression analysis of both protein and mRNA detection. So building upon the GeoMx system really built upon a lot of the foundational work that was done with our nCounter system to look at multiplexed expression signatures. So this is a study we had done with Merck where we had looked at a predictive value of gene expression signatures on a bulk level for head and neck cancer with their checkpoint inhibitor therapy, Keytruda. So in this case, looking at an 18 gene inflammatory signature that was identified as part of this study, they were actually able to improve upon the predictive power for this drug over a single stain PD-L1 IHC, which is at the time the current gold standard. So what we were able to prove with studies like this is the value of expression signatures as predictive tools for response to treatment or for potential biomarkers. Now, as great as this study was for enriching for a population that could potentially benefit from this checkpoint therapy, not all patients who had high inflammatory scores, which in the chart you see at the bottom there is categorized as GEP or gene expression profiling score, not all highly inflamed tissues that receive this high inflammatory score responded positively to this checkpoint therapy. So all of the potential and excitement around these checkpoint therapies and rightfully deserved there's still a lot of room for improvement upon improving predictive power of who should receive these treatments and how they'll fare once receiving that treatment. So bulk expression profiling has gotten us so far. Once again, we can enrich for a population that could potentially benefit. But when we think about things like adverse side effects or the cost of these treatments, the need is there for better predictive tools to really narrow in on that precision medicine goal of giving the right treatment to the right patient at the right time. So one of the reasons, these bulk signatures, can give us a great predictive power, but maybe not enough, is that they're not taking things like spatial heterogeneity into account. So while we look at two separate samples here, both with what we would consider high inflammatory scores, right? There's a lot of immune cells present within the sample on a bulk level. When we look at them spatially, we can actually see they're quite different. On the left-hand side, we're seeing an exclusion of those immune cells from the epithelial cells or from what would be considered the tumor. And on the right-hand side, we're seeing high infiltration of those immune cells directly into those epithelial cells. So in this case, if we're going to do something like a checkpoint therapy where we're removing those breaks from the immune system to be able to go out there and attack those tumor cells, we really want to make sure that those immune cells are actually infiltrating and primed into the tumor itself before receiving that treatment. So once again, bulk tools have gotten us so far, but also being able to look at a spatial level can provide further granularity into what's happening within things like the tumor microenvironment that might help lend itself to a better outcome for treatment. Now, the challenge historically has been that spatial tools have been fairly low plex. So things like immunohistochemistry or immunofluorescence even can only look at a handful of targets at a single time. They're fairly qualitative in their approach for the most part. Now we have ways of getting closer to digital pathology by doing things like optical counting or analog to digital conversion. But ideally, we want to be able to get as much of the biology as we need to understand to really understand the complexity of something like a tumor microenvironment. But we want to be able to do that from a single tissue section, so from as little sample input as possible. So we know how valuable and precious clinical samples can be. So if you really want to get at all of the underlying biology of something as complex as the tumor microenvironment, you wind up with lower plex technologies, you wind up taking lots and lots of serial sections to do that. And once again, with limited sample, that may not be available to you. Where ideally, with something like our GeoMx system, we want to get all of this biology out of as little sample as possible. So ideally from a single FFPE tissue section, and getting all of the combinatorial information that's gained from using just that one single section, as opposed to the biology that changes as you go further and further into that block. So that's really why we've developed the GeoMx system. It really incorporates now the spatial information from things like immunohistochemistry and immunofluorescence with the highly multiplex quantitative profiling power of things like our nCounter system or even next-gen sequencing. So we're now, with the GeoMx platform, building this bridge between being able to look at the spatial information and then attach that or combine it with that high-plex characterization and profiling. And once again, as a translational tool, it's extremely valuable and important that it works within the tissue samples that are available to most clinical labs. So things like cord needle biopsies, FFPE tissue blocks, maybe pre-mounted tissue sections, but even things like fresh frozen tissues or tissue microarrays. The GeoMx system really enables you to go back and look at these samples that you have access to and do that deep characterization. So both for prospective as well as retrospective studies. And as a multi-omic tool, I mentioned this is a multi-omic platform for detecting both protein and mRNA. We've developed different chemistries and different probe sets for getting at those different analytes. So on the protein side, it is an antibody-based chemistry. So these antibodies will recognize and bind to your target proteins, and attached to those antibodies is a synthetic DNA oligo tag. So this is a single-stranded synthetic DNA oligo tag that attaches to that antibody, and we attach that oligo via this UV photocleavable linker. So we can actually take a cocktail of these antibodies, and really the limiting factors of the amount of plex that we can do is our ability to read out those tags. So in the case of our nCounter system, theoretic limit is up to 800 plex. In the case of Illumina next-gen sequencing, when we want to actually sequence those barcode tags out, we can theoretically go entire proteome or entire transcriptome. There's really no limit to the amount of plexing that we can do with the platform currently. So that's protein detection is antibody-based. On the mRNA side, we're doing an in situ hybridization. So this is an ISH chemistry that will hybridize to the target RNA in situ, and then attached to that hybridization probe is a photocleavable linker with, once again, that synthetic DNA oligo tag barcode attached to the photocleavable linker. Now, the photocleavable linkers are extremely important because what we can do is stain up our tissue section. So say you have a 5 micron thick FFPE tissue section. You can stain up that section with this cocktail of probes and antibodies. Identify regions that you want to profile within the tissue using just a 3 to 4 plex immunofluorescence stain that's spiked into the slide itself. Identify through fluorescence the regions of the tissue that you want to characterize and profile based off of their morphology. And then what the system does is it moves the objective field over the first region of interest that you've selected, turns the UV light on directly through that objective. There's a UV illumination built right into the microscope, so the UV light goes right through the objective. And what that will do is it will release that photocleavable linker, releasing those indexing oligos, those synthetic indexing oligos, into a liquid that's sitting atop of the tissue. And then a microcapillary system can come through and sip up those oligos from each region and dispense the oligos that were released from each of those regions into a 96 well microtiter plate, where each well on the microtiter plate that has those photocleaved oligos in them can be indexed back to where it came from spatially back onto the tissue. So in the case of the tonsil section that you see on the right-hand side here, we did an immunofluorescence stain. So what you're seeing in those 3 color stains is just standard immunofluorescence. We did a pan-cytokeratin in green. It's a CD20 in yellow and a CD3 in purple. So we've selected germinal centers, T-cell beds, and epithelial layer. We went through, released those oligo tags from each of those morphological regions, and then profiled an expression panel of, I think in this case, it was about 30 to 40 proteins from each of those distinct regions individually, and then map that expression back to where they came from on the tissue. So you can see the ROIs that were selected on the tonsil on the right-hand side, and on the bottom, you can actually see the heat map from the expression profiles from each of those individual regions. So we can see clustering as we would expect, expression profiles matching up with the different morphological structures, but then we can also do a deeper level analysis of actual expression gradients and expression profiling as we go across the tissue as well. This protocol, at least the antigen retrieval and staining, has been automated. So we have a partnership with Leica to automate the staining and sample prep on their Bond RX system. And the readout of those oligo tags, as I mentioned, can either be done on our nCounter system, but we also have a partnership with Illumina that allows us to read those oligo tags out on their Illumina next-gen sequencing platform. So this is just a quick concordance study. The first things that folks want to do once they get their hands on the GeoMx system is do a quick concordance with their IHC scoring. So they've got their spatial data from maybe some lower single or multiplexed IHC, and they want to compare their single or multiplexed IHC scoring, pathology scoring, to the expression level data that's coming off of the GeoMx system just to make sure it's matching up. So one such study was a study we had done with Josh Roosevelt, who's a scientist in the translational sciences translational research team at Janssen Research and Development in Springhouse, Pennsylvania. And as part of our initial study with them, we looked at some 5 non-small cell lung cancer tumors where we had done where they had previously done some IHC scoring for a handful of targets, CD3, CD4, CD8, CD68, PD-L1. So a high, medium, low scoring for those targets. And then we went through and actually digitally profiled those same targets for each of the same morphological regions within those same tissue samples. And what we found was great concordance. This is a study that we've presented previously, and it's now available on our website for download, where you can actually look at the comparison data between the DSP digital counts as well as the pathology scoring. And one of the great things about this platform with the large dynamic range that we have is you can actually see where there's discrepancies in expression even within single categories for the pathology scoring. So in this case, looking at IHC scoring in that high category for CD4, we can actually see variation in expression level of CD4, where the kind of standard pathology scoring would really just kind of bin all of those things together. So once again, the value not only in the multiplex, but also in the digital capabilities and the large dynamic range that the system offers. We have a really neat device that we built into the GeoMx microscope itself that allows us to do mapping of morphological structures and cell types. So it allows us to do compartment-based profiling directly on the tissue section, similar to what you might do with like a laser capture microdissection, but without actually physically destroying the tissue. So all of the tissue is still intact after we do this profiling. So in this case, that's a colorectal cancer that you see in the middle there. There's a pancytokeratin stain in yellow. So all of those epithelial cells staining positive in yellow for pan-CK. And those would be our tumor cells in yellow. What we're able to do is with an image analysis algorithm that we've built into the microscope directly, we can set a gating criteria for tumor cells. So basically anything pan-CK positive that we want to profile, that becomes that mask that you see in the top right-hand corner there. So everything in white would be all of our tumor cells. That mask then reconfigures this digital micromirror device that we've built into the microscope, which allows us to control the UV light in these little 1 micron increments. So it allows us to shine UV light in that core colorectal cancer only on the tumor cells, which means we're only going to release the oligo tags from the bound probes or antibodies on those tumor cells and nothing around the tumor cells. So it allows us to actually profile a large multiplexed panel with digital expression on all of our tumor cells in the tumor compartment. It's a binary mask. So as an inverse, we can also take anything that's pan-CK negative and make that a separate region for profiling. So we can do both tumor and tumor microenvironment completely separately, profiling in extremely high plex using the system without ever physically destroying the tissue itself. And so this is just an example. This is once again a colorectal cancer where we went through and in region 1 selected a region that we wanted to profile, set a gating criteria for tumor versus stroma, and then profile tumor versus stroma completely independently of each other. Once we set that gating criteria on the tissue section in that first region, we can then drop subsequent other regions across the tissue, and the same gating criteria will follow through. So we can look at things like the invasive margin, the tumor center, and even normal colon, but we can now do that by profiling just the tumor and just the stroma cells independently of each other. And that's the heat map that you see on the right-hand side there. So this ability to look at signatures within individual compartments. And so I've shown you some fairly targeted work that we started with early on with our GeoMx system in the early days. So back in 2019 when we launched, we were able to do about 84 targets at a single time. And you can see how rapidly we've been able to scale the amount of plex that this system can handle. So in 2019, 84 genes was kind of the standard of what we were able to do. In 2020, we launched our Cancer Transcriptome Atlas. So this is an 1,800 gene curated panel that allows you to look at 1,800 genes that have been curated for cancer biology, looking at the immune and tumor response, as well as specific pathways associated with cancer progression. And then most recently now in 2021, we enabled whole transcriptome profiling on the GeoMx platform. So we have both human and mouse whole transcriptome capabilities, anywhere between 18,000 and 22,000 genes enabled directly on the GeoMx system, all from a single 5 micron FFPE tissue section. So really exciting times. And once again, this is now on the mRNA side where we've really been able to scale up. On the protein side, the most plexing we're doing right now is about 140 plex. But certainly no limit to the amount of plexing we can do on the protein side either. So we're very excited to continue to push those boundaries, both for protein and mRNA on the platform. And so this is just showing you once again with that masking capability, being able to look at both protein and mRNA detection in a single study, looking between tumor and tumor microenvironment. And in this case, we were comparing our Cancer Transcriptome Atlas with our human protein cores for looking at patient samples that were both on and off treatment to do differential expression to understand how the protein and the mRNA expression differs from patients both on and off treatment going across both the tumor and the tumor microenvironment. So really nice digital expression data, no pixel counting necessary, gives you that really nice quick digital readout, which can be very valuable for these translational studies where we're trying to do differential expression between different populations of patients or samples. So I will summarize really quickly. The GeoMx system, once again, extremely powerful, allows you to detect all the targets you're interested in, both protein and mRNA, with high sensitivity and a large amount of plex. It allows you to work within the regions you're interested in, so that combination of IF with that expression profiling allows you to select distinct regions and morphologies that you're interested in. So you can really do the research and answer the questions that are specific to your study. And it allows you to work with samples that are available to you in the lab. So both fresh frozen and FFPE, as well as TMAs, core needle biopsies. So with that said, we have a few polling questions that I'm really excited to get your responses on. This will help define and further evaluate our portfolio. So we're looking to get you to respond to some of these questions. So please, a poll will open up and you'll see the questions on there. And I'm happy to read through them as well. The first polling question is, what is the ideal plex for spatial protein detection? I mentioned, we started off with very targeted work. We're now up to whole transcriptome, but specifically for protein work, what is the ideal plex for the research that you're doing in your lab today? So, 0 to 20 for option A, 20 to 50 for option B, 50 to 100 plex for option C, 100 to 500 plex for option D, or 500 to 1,000 plex for option E. So I'll give you just a quick second. If the poll is open, please feel free to respond, and then we'll move on to the next question. Oh. I think, okay. The next question is, what analytes do you prefer? I mentioned this is a multi-omic platform, but we'd love to get your feedback as far as what in your lab specifically, the type of work that you do, whether it's protein, RNA, or maybe it's both. Maybe it's multi-omics, the relationship between protein and mRNA. So we'd love to get your response on that. Okay. One more question for you, or I think we have a few more quick questions. What is your most common sample type that you're working with in the lab? Is it FFPE? Is it fresh frozen, fixed frozen? Are you working with tissue microarrays in the lab? Or is it some other format that maybe I didn't mention here? Great. And then I think this is our last question is, do you have access to an autostainer in your lab? So I mentioned our partnership with Leica on their BondRx, but we'd love to hear if there's other autostainers that you're working with today, what those might be. So Leica for A, Thermo Fisher for option B, maybe it's Daco for option C, or Ventana for option D. And of course, if you're not working with an autostainer in the lab or you don't have access to one, or if there's some other option, feel free to select option E. Great. Well, I hope you enjoyed the introduction here, and I will hand it over to Dr. Chris Corless, who is the director of the Knight Diagnostic Laboratories at Oregon Health and Science University. And he'll take you through some of the work that his lab is doing to evaluate the GeoMx system across their breast cancer samples for predictive or for biomarkers.
Great, thank you so much, Nick. It's a pleasure to have a chance to participate in this forum. Thank you everyone for joining. So the work I'm going to discuss today is focused on proteins. Both our group and the Mayo Clinic, and I'm representing both groups in this presentation, are interested in doing RNA work, and we're beginning to go down that road, but today I'm just going to be focusing on protein work. So this is a collaboration that was started a little over a year ago between Mayo Clinic, our laboratory at the Knight Cancer Institute, and then Nanostring, and I've listed the authors here. So the goal was to develop a breast cancer biomarker panel and to test it out to see whether or not it could perform to the level that we would want in a clinical setting. So with that as a background, just let me outline quickly what I'm going to share with you today. I'm first going to show you a research example of how we're using the DSP platform routinely in the laboratory. Then I'll go into a little bit more detail about how we developed the breast cancer biomarker panel. I'll show you a little bit of preliminary data that we've generated from that panel. And then I'm going to come back at the end and talk in detail about one set of biopsies from a clinical trial to illustrate how you can use this platform to complement other technologies and learn more from the tissue and get a better understanding of what's happening in a clinical trial. So let me begin then with an example of the use of a TMA, so a tissue microarray that we just analyzed very recently, and we did all this work in less than a week. This tissue microarray consisted of 88 breast cancers and some cell lines that were paraffin embedded. This work was performed as part of our work for the Human Tumor Atlas Network, so OHSU is one of the participating institutions there. So as part of that study, we went ahead and analyzed this entire group of core biopsies that were represented across these two slides by doing the following. We collected signals from CD45 positive cells as segment 1, and those are the cells shown here in blue. Then we collected signals from the pan-CK positive tumor cells, which are the green guys dominating this region of interest. And then the background stromal material that's left over, that was the third collection in segment 3. So we did all 3 segments across all of these cores. And this is the unsupervised cluster analysis of 87 antibodies run across that tissue microarray. And you can see in green are the areas, the region of interest that were focused on the CK positive tumor cells. In red are the CD45 populations, and then the background color here is just the stromal population. And what's nice is that you can see right away that the tumor populations nicely distribute themselves into recognizable groups. So for example, over here on the right, we have a group of tumors that have very little in the way of estrogen or progesterone expression, but have lots of HER2. So these are HER2 basal type tumors. Here in the middle section, we have this luminal group, strong expression for the hormone receptors. Interestingly, many of them showing higher expression of cyclin D1. And this is all consistent with a luminal subtype of breast cancer. And then over here, we have a group that we could classify as basal-like, and they're characterized by having a relatively high c-MYC expression in many cases, overexpression of EGFR and phospho-EGFR relative to the luminal tumors. And interestingly, many of them showing very strong signals for P53, which is fully expected because most of the TNBCs or basal-like tumors will have P53 mutations. And so the immune cells are showing appropriate signals for the T cell markers as well as the macrophage markers. The stromal collections are dominated by fibronectin and smooth muscle actin as expected. So this is just a quick overview of the kind of data that you can generate in just a few days from tissue microarrays. Now, in working on the development of the breast cancer panel, this is a project that actually started about a year ago, a little over a year ago. And interesting how you can conduct work like this during a pandemic. But what we did together with our colleagues at Mayo Clinic and at Nanostring was to first decide on what antibodies we wanted to have in a custom panel, then to purchase them and individually screen them. And this was done primarily at our group by immunohistochemistry or in some cases immunofluorescence, just to make sure that the antibodies that we were selecting, commercially available, were performing as expected. And then once we had chosen the group of antibodies, they were sent to Nanostring and were tagged with the oligonucleotide linker, the little marker that is used for the quantitation. That was then sent back to our group, and the post-tagged antibody was reevaluated to make sure it still had activity. And then finally, we completed a multiplex of all the antibodies together and distributed that for testing at 2 different sites in Mayo Clinic and in our own laboratory. Right now, we're doing multi-site and intra-site concordance studies, and we're doing a bunch of other studies, which I'll illustrate for you in a moment. So, but there are many different aspects that are important when you're trying to look at antibody performance. Nick touched on this already a little bit, but I just want to emphasize that especially if you're thinking about using something in the clinical realm, you need to put the antibodies through their paces, and that means looking at every possible orthogonal check that you can. In this case, we have a set of 9 standard cancer cell lines, many of them breast cancer cell lines, that were embedded and formalin fixed into a tissue microarray. And then these cell lines have already been analyzed by reverse phase protein array at the MD Anderson core. That data is publicly available. So we compared the DSP signals that we got for P53, ER, PR, and so forth with the data that were seen by our PPA approach. And the correlations really are quite striking across these 9 cell lines. So when you see data like this, you say, okay, the antibodies are performing as expected, and we have a nice orthogonal check. And of course, we're also doing IHC and IF, as I already mentioned. We're comparing against RNA sequencing data that's available either publicly or through our own work. We're looking at genomically annotated cell lines as well as tumor samples where we know that one gene has been mutated, so that should be overactive, or there's been a loss of function mutation. And we're also looking at cell lines that have been treated with drugs or growth factors where we can try to goose the signal one way or another and make sure that we're getting appropriate changes in signal for each of the antibodies in question. So it's a combination of these approaches that then allows you to build a collection of antibodies in which you have full faith that you are getting the appropriate signals. Here's some pilot data from our group. It's now being replicated as part of the Mayo collaboration. Here you see hormone receptor positive tumors showing strong ER signal on the DSP, and triple negative tumors basically undetectable. Here you see the spectrum of HER2 signals across varying levels of staining, and even 2+ FISH positive tumors showing a slightly higher signal level than just the general 2+ tumors. So this kind of trend is the kind of thing that gives you heart that the antibody is performing well. And in fact, and again, Nick already alluded to this, allows you then to draw a line and then put your individual tumor right on that line, rather than putting the tumors into these rather arbitrary bins, the traditional bins that are used by pathologists. We will be generating a lot more N values for both ER, HER2, and a number of other markers as part of the Mayo Clinic study, and that data will be published in the next quarter. Here's some other examples of genomic correlations that we've done. This is a group of tumors that have normal P10 based on DNA sequencing, and you see the level of signals that we get on the DSP. Here are 12 tumors that had either loss of function mutations or deletion of P10, and they all have significantly lower signals. And then over here, we have P53 signals across a variety of tumors. The group on the left all have P53 point mutations, and we would expect to see overexpression of P53 under those circumstances. That's well documented in the literature, and that's exactly what we see as compared with normals over here. And then on the right side, we see a group of tumors that have either truncating mutations or splice site mutations, where we would expect to see very little P53 expression, and that's exactly what we get. So it's kind of fun, because once you have gone through these kinds of exercises, you can literally genotype a tumor by just looking at the protein expression levels. And that it's satisfying in many ways to have that additional check on the tumor. You get a certain level of understanding from the DNA, but sometimes it's not quite clear whether a mutation is really dominating or whether loss of function mutation is high enough that you'll have complete loss of the protein. And the DSP allows you to check that very nicely. Now, turning our attention back to the breast cancer biomarker panel, that's what we have shown here. This is the final list. It was intended to be a little bit bigger than this, but some of the antibodies fell out during our screening process, and so we did not include them. On the left, we have the 5 traditional clinical markers used by pathologists in assessing breast cancers routinely. We have some additional drivers, including phospho-HER2 and phospho-EGFR that we've included here. Some breast cancers have FGFR1 amplification, so we're interested in measuring that. We also have these cell cycle markers, immune markers, not a lot of them as compared to the ones that are commercially available. If you wanted to do a deeper dive of immune markers, you would use one of the other modules. But here we just wanted a smattering of different common markers to assess the level T cells and macrophages that might be present in a tumor. And we have a couple of other targets over here as well. Now, one thing that I think is worth pointing out is that if one is going to be using phospho-specific antibodies, they require additional vetting, and we've been doing that by, for example, pre-treating slides with phosphatase and showing that the signal goes away. But in addition, you have to be aware that if you're going to evaluate those antibodies, then it can only be done on tissue that has been properly fixed. That is to say, within minutes of being removed from the patient. That eliminates a lot of specimens, and we're well aware of that. And the intention of the intent for the use of this particular panel is on core biopsies that have been fixed very quickly. And that is often the case where a patient is undergoing a core biopsy under ultrasound guidance, and then that core is going into formalin within a couple minutes. That's the kind of specimen where we can use these phosphomarkers, but a lumpectomy or a mastectomy specimen might not be appropriate for some of these markers, just to keep that in mind. Now, we built a tissue microarray that we're using now as a standard control for the entire set of experiments. And the elements of that tissue TMA are shown right here. Many of them are embedded cell lines that have either been treated as controls or with things like CDK4/6 inhibitor or with a growth factor like neuregulin or EGF. And the idea then is to get a complete spectrum of signals that allows you to assess antibody performance, not just during initial screening when we're establishing the panel, but also as positive and negative controls on every run. So we evaluate all of these spots as part of every single run, and it also includes some normal tissues like liver, tonsil, and spleen. The other advantage of doing this is that this now gives you a benchmark for doing data normalization between runs. In our laboratory, we're often in a situation where we have a patient's biopsy in April that we want to get analyzed right away. And then the patient comes back 4 months later and has another biopsy, and we want to analyze that, but it's going to be run on a different day with different conditions in terms of temperature and humidity and all the other minor things that can influence an experiment. So by having the TMA present on both of those runs, we can now make changes in the data normalization strategy so that any slight changes in signal can be taken out of the data, and then you can do direct head-to-head comparison. Okay, so for the Mayo study, we've been looking at concordance, and intra-laboratory concordance is being done at Mayo Rochester. So here we have run 1 and run 2 for a core biopsy evaluating 4 different ROIs. You can see that ROIs are being selected very carefully to match between these 2 adjacent cuts. And then this is allowing us to generate data for the one laboratory reproducibility day to day. But we've also had these same sections sent around to the other 2 sites, Jacksonville, as well as our laboratory, and are evaluating them in the same way. So just to share with you some of the data, so this is the intra-laboratory reproducibility, run 1, run 2, done at Mayo Rochester. Beautiful reproducibility of these markers. I mean, this is the kind of thing that really gives you great confidence that this platform is truly quantitative and robust. Now here are the data showing the concordance between the 3 sites. And you might be a little bit more alarmed because there's more scatter, but what's happening here is that 2 of the sites show great alignment, the orange and blue sites, and then the 3rd site, the green site, is showing consistently higher signals. And it turns out that there was a slight protocol deviation that happened at that site. It was an accidental thing, but what's interesting is that the signals overall are parallel, they just happen to be higher. And so in some ways, the data is still useful. But you just have to be aware that when you're conducting studies like this, you have to be very precise about the protocol, and we are, of course, going to rerun this experiment. But I'm sharing this with you because, I mean, these are real life data, and these are the kinds of things that happen when you're working between institutions. Nevertheless, we have confidence that this system is robust. So some of the ongoing studies that we're doing with Mayo, the standard breast markers are all going to be compared DSP versus digitally quantitated IHC. Ki-67 is going to be looked at in comparison with standard MIB1 IHC as well as with Oncotype DX results. We're looking at immune markers for tumor infiltrating lymphocytes in triple negative tumors. We're looking at PD-L1 performance. We're looking at TROP2 versus response to sacituzumab. It'll be interesting to see whether there's any correlation there. Some data in the literature suggests that there's not, but we'd like to go ahead and take another look at that. And we're also going to be looking at a group of xenografts grown at Mayo Rochester that have been treated with either control or with CDK4/6 inhibitor to see how those look. And so all of these data are being generated right now. In fact, most of it's already been generated, and we're doing a lot of data analysis, and then we'll be submitting a publication in the next quarter. So in the remaining time, what I'd like to do is turn our attention back to a clinical trial that's being done at our institution. We're deploying the DSP platform for a number of clinical trials, most of them on breast cancer, but also for ovarian cancer, pancreatic cancer. We've been very pleased with the performance. Now, all of the antibodies here are the ones that we're running for this particular trial, and among these, there are a group highlighted in red that are not commercially available. These are antibodies that we purchased and then had custom tagged by Nanostring. It went very well. These antibodies are performing nicely. All the rest of everything that's shown here on the slide is commercially available from Nanostring right now. So we've run this entire cohort on a couple of different clinical trials, and I want to share with you one set of biopsies from a current trial. On the left, we have the pre-treatment core biopsy, and this is a closer up view of the tumor here. And on the right, we have the on-treatment. So this is 4 weeks in on treatment with triple therapy. Now, in this case, the patient's being treated with a CDK4/6 inhibitor, a HER2 EGFR inhibitor, and an aromatase inhibitor. And so the interest here is that this is being done as part of a neoadjuvant approach. In other words, this is a newly diagnosed breast carcinoma. This is the original biopsy. This is 4 weeks in on this pre-surgical therapy, and then the patient will be undergoing either lumpectomy or mastectomy in a few weeks' time. So we're interested in asking the question, what's going on in this biopsy under the influence of these drugs as compared to before treatment? Morphologically, I'll point out right away, there's not a big difference here. The tumor cells appear to be still healthy. There's still a strong lymphocytic infiltrate, much higher than we typically see in a breast cancer, but it's also present in the pre-treatment sample as well. So not a big difference, so keep that in mind. Now, what we did in this case then is we collected antibody signals from the tumor cells, so shown here, and then we went back and collected a 25 micron zone around these tumor cells to get at the immune signals that might be at the interface between the stroma and the tumor. And by the way, these lines here were drawn by me and my mouse on PowerPoint, and not from the actual instrument. So don't take these lines too literally, please. Now, these are the data, and the way that we plot the data initially is using box plots. And we're comparing each biopsy with 20 other breast carcinomas that we previously analyzed with the same markers. 9 of them were TNBCs, the rest were hormone receptor positives with a wide expression level of HER2. Now, from this, we can then determine what is the percentile of the current sample relative to these 20 other breast carcinomas. And we did this both for the pre-treatment biopsy and the on-treatment biopsy. Now, looking a little bit more deeply at this pair of biopsies that I'm showing you, we did sequencing on both of them. There was no change in the DNA sequence between the two biopsies. There is the presence of 2 different PI3 kinase activating mutations. And so what we would expect to see is high signaling in the PI3 kinase cascade. There's also a truncating mutation in a RID1A, so we'd expect maybe that level would go down. But what was really interesting is that this tumor has a lot of mutations overall, and this tumor mutation burden of over 30 is very high for breast carcinoma, which is in perfect keeping with the fact that there is such a dense lymphoid infiltrate in the tumor. Typically, the tumors that have more neoantigens as a result of mutations are the ones that garner a higher, lymphoid response, and that's exactly what we're seeing in this tumor. Okay, so looking at the DSP results, there was really no change in HER2 levels. It was 2+ by IHC, FISH negative, and it's 45th percentile versus 75th percentile pre and on treatment. Not much difference in ER levels. Ki-67 was low by IHC and at the lower limit for the GeoMx, and it stays low on treatment. Now, but looking at some of the other markers is quite interesting. I pointed out that there are these activating mutations in PI3 kinase, and lo and behold, we have very high signals for phospho-AKT, phospho-Pras40, and one of these phosphatases in the pathway, and it's present both pre-treatment and still quite persistent on treatment. Now that makes sense because none of the 3 drugs here is targeting PI3 kinase, so we don't necessarily expect to see a big drop in PI3 kinase signaling. Now, when we look at ARID1A, it was undetectable pre-treatment. It's still low on treatment. That's consistent with this loss of function mutation. But here's where it gets really interesting. When we looked at the cyclins, cyclin D was low in both biopsies, but cyclin E was at the 70th percentile pre-treatment and not detectable on treatment. And that's exactly what we would expect from a CDK4/6 inhibitor. We would expect that the cell cycle would basically be shut down and the cyclin E levels would drop, and that's what's happened. What's sobering then is that this is telling us that we are probably getting the target that we wanted to get with the abemaciclib, but it's not really having much effect on the tumor. I mean, the tumor still looks like it's healthy. There's no necrosis, really no other overall much change. The apoptosis marker shown here didn't really change. Phospho-HER2 is the same. Cleave caspase was going down a little bit, and STING was going up a little bit. Whether these changes are important remains to be determined. But anyway, this is a nice example of how you can use this kind of technology to evaluate clinical trial samples and get quantitative readouts that provide meaningful insights into what's happening in the tumor. This particular tumor may not be benefiting, or I should say the patient's not necessarily benefiting from this particular therapy, but we can learn from that. And we can go on and try different therapies in the future. In terms of the stromal measurements, not much change in most of the markers, but TIM3 went from undetectable up to the 85th percentile in the on-treatment biopsy. Perhaps there's been some influence by the treatment, and there's more T-cell exhaustion in the on-treatment setting. Again, this is just one measurement, but it's kind of interesting, and it's the kind of thing that we will be looking at as we look at more biopsies going down the road. So to summarize then, what I've shared with you, I hope, is that this is a very powerful system that will allow you to look at least 90 protein targets within one tissue section. If you switch over to the NGS readout, which we've not done yet, but if you use the NGS readout, there's really no limit to the number of protein targets that you could approach. It's a single section that's required. 48-hour turnaround is possible. We're generally running 48 to 72 hours. And we're finding that the markers on the breast cancer biomarker panel are performing well in terms of our early reproducibility studies, and we think that they will be very robust for future breast studies, in particular for clinical trial analyses. And I've shown you an example of how this can be impactful, and we think that deploying this going forward on more trials, and we have quite a number of them coming up in the next year, will give us that additional omic element that we've been missing by just looking at DNA and RNA sequencing on those samples. So with that, I would thank you for your attention, and I'll be happy to take questions.
Thank you, Chris. That was really an incredible presentation. We did have a question come in, and it sort of speaks to your last slide, and that was how high of a plex, or what is the level of plex that you envision using for future diagnostics?
Yeah, so I think that you have to ask the question, are you going to be using it in routine clinical practice, or are you going to be using it for more research-focused, clinical trial type applications? For routine practice, I think a smaller panel would be appropriate. The breast cancer panel that we put forward was really intended for that. So it's around 24 or 26 markers, and that's probably a pretty good size. We've kind of sketched out what a GI panel might look like, and that would be probably in a similar range of between 1 and 3 dozen markers. And so the beauty of it, though, is that if you do that, you only require one section to get all of the data from those markers. And that leaves a lot of material left over in the block to do the other kinds of things that people like to do. One of the great frustrations I have in our laboratories here is we get blocks every week that are sent to us that are completely exhausted. And I don't want to point fingers, but the pathologist who was doing the original diagnosis ran 15 or 20 immunohistochemical stains and then said, we need to do DNA studies. Well, there's no tumor left. So I think that having panels that bring forth the key markers, a dozen, 2 dozen, whatever it might be, would be very helpful in a clinical setting. And then if you're doing research studies, you might want to ramp it up. And like I said, we're running around 90 markers, and I think there's interest in doing a lot more of that in the future.
Excellent. That's very helpful. There was a question here about how are you managing data analysis?
So the software that comes with the system allows you to do a number of different types of analyses. A lot of it is built on geomean normalization of the data. In our case, we have added on the tissue microarray as a marker for every batch. And that allows us to make sure that all the antibodies are performing as expected on every batch, and then to do normalization between batches. Those extra steps have required considerable input from our own bioinformaticists on campus, but we will be publishing our strategy in the future, and it's something that other people could adopt if they want to.
That's great. I think that was exactly what the question was getting at, is can we mimic what you've accomplished already? I see we're over time. I'd like to thank everyone for your time and attendance, and I'd certainly like to thank our speakers, Nick Conforto and Dr. Christopher Corless, for their time and energy putting together this presentation and sharing the details of their project. So thank you very much, and have a good day.